So we better get started here. Welcome to the last session of today and of the conference. This session is invasions and inventories, and we have 4 great speakers. So it's good to see almost a full house here. Our first speaker is Andrea, and she's going to talk to us about garter snakes in Newfoundland. Go ahead, Andrea.
Awesome. Thank you. So this is kind of an update on what's going on with the invasive garter snakes in Newfoundland. I've presented on this sort of a bit, but not everybody's seen it yet, so we're going to go over everything. So just to sort of orient us, with invasive species, they're not great for biodiversity. So invasive species cause a lot of extinctions, and they do this by increasing predation pressure and competition for native species, and thereby disrupting ecosystem function. So a great example of a species that does both of these things is Burmese pythons in Florida, which both eat basically everything that they can, and then they also compete with other sort of meso and almost like major predators for resources as well. So that said, invasive species is really bad, but not every species that's introduced to an area actually becomes invasive. So a bunch of things have to happen for a species to go from introduced to invasion. So first, obviously, they have to be introduced to a new area. The first thing that they have to do after that is actually survive in the environment. If they can't do that, whomp, they fail. If they can, their next challenge is that they actually have to be able to reproduce in the environment. So if you just have one individual, it's not really going to become an invasive species. If it can't make more of itself, whomp, doesn't make it. But if it can reproduce, the next challenge that it actually has to do is spread past that point of initial introduction. If it can't do that, guess what happens? Whomp, failure. But if it can, then it's actually considered an invasive species. So there's a lot of characteristics that make it more likely for an introduced species to actually overcome all those hurdles and go all the way to becoming invasive. And I put a picture of my cat here. Her name is Newt, because cats are really good at becoming invasive species. And we'll just go through some of the characteristics that make species more likely to invade. So one of the biggest ones is climatic match between native and invasive range. So there's no cats in Antarctica. They can't hack it. There probably would be if they could. Association with human populations. So cats are really good at this. We really like them, and we bring them everywhere with us, which helps a lot in the invasion process. High propagule pressure, hashtag. That means in the invasion literature, that refers to the number of individuals that are introduced to a single area. So a lot of individuals being introduced at one time, or a sort of steady trickle of individuals being introduced over time. And the more individuals that are introduced to an area, the higher the likelihood that they're going to be able to reproduce and spread. And of course, cats, we bring them everywhere in large numbers. High vigility, so this means their ability to travel through the environment. So that really helps with the spread phase of becoming invasive. And if you've ever been hunting in the woods in New Brunswick, really far from human habitation, you might still find cats out there. So they're very good at going really far. High fecundity, so generally the number of offspring per year or per lifespan of the individual, depending on what you're looking at. So cats, if you know, they can reproduce really early and really often. Flexible habitat use, so habitat generalists are better able to become invasive than habitat specialists. The same goes for diet, so we've got our generalist carnivores are going to do a lot better than a specialist feeder. Behavioral plasticity, so the ability to adjust their behavior really helps, and cats are very good at that. And then behavioral traits like boldness and exploratory tendency. And when you have all these things and you have cats that are introduced, you go from a cute cat to a scary feral cat. And these are the ones that are really devastating and causing a lot of problems. So, Newfoundland. Newfoundland is historically snake-free. It's actually naturally free of any herpetofauna whatsoever. But in 1974, there was a snake that was found in Kidividi, which I've indicated there with a little snake icon. And then kind of nothing. They laid low for a really long time in the official reports. But in 2010, there was a snake found breeding in Western Newfoundland in the community of St. David's, which I've indicated over on the West Coast. In 2015, there was another snake found in Southlands, which is a suburb of St. John's. So that's in the sort of same area as Kidividi over on your right. And then in 2018, there was a snake reported in Middle Arm, which is kind of up on the northern part of the island. Stopped a butcher in his tracks. So this is 4 observations over about almost 50 years. So does that mean that there's actually sustaining snake populations on the island, or are these just kind of one-offs? So to kind of dig into that, I made a Facebook group called Newfoundland Snake Sightings, which now has over 5,000 members. And I asked people, have you ever seen a snake in Newfoundland? And the answer was overwhelmingly yes. So each one of these dots on this map indicates either a whole animal, and those are the orange, yellow dots on this map. Or actually, each of these dots represents a different community in Newfoundland. So the orange ones are places where we found a whole animal. So those range from animals that were picked up by the Wildlife Division, animals that were found by people as roadkill and kept in their freezer, and then animals that I collected in 2023. There are also photographs. So the blue dots represent photographs that were submitted to me with a date and a location. And then the verbal reports, which are the gray dots, are people who reported that they or somebody that they knew had seen a snake at some point between now and the dawn of time. So there's quite a spread in how verifiable that data was. So in 2023, I got in a little rental car and I went to Newfoundland and I visited every single community on this map and more, and asked people about the snakes. I said, have you ever seen a snake? Have you ever heard of anybody that's seen a snake? Where have you seen a snake? And through doing that, I found snakes in these 3 areas. So the lowest one, the farthest south is St. David's Robinson's area. The next one kind of in the middle is the community of Trout River. And then up on the northern area here is the community of Middle Arm. So why are we concerned about this? It's because garter snakes could potentially be good invaders because they tick quite a few of those boxes of characteristics that make good invaders. So for one, they have a lot of offspring. I actually got a litter given to me from the Wildlife Division that was 38 neonates from a single female. They are habitat generalists. They are generalist carnivores. There's also a good climatic match between parts of their range and Newfoundland. What we don't know is how many individuals were introduced either at a single time or whether there's been continual introduction of individuals over time. We don't know how plastic their behavior is. And we don't know if they exhibit these specific traits like boldness and exploratory tendency. So my mission, which I've chosen to accept, is to find the source population or populations for garter snakes in Newfoundland, determine behavioral differences between that source population and the invasive populations, and then predict potential ecological impacts of garter snakes in Newfoundland. And this is what I'm focused on right now. So this is the only thing that I'm thinking about at this time. So to do that, this year I went back to these 3 communities and I collected as many snakes as I could find. And in Middle Arm, this year we collected 24 snakes. We collected 11 in Trout River, and we collected a whopping 89 snakes in the Bay St. George South region, which covers St. David's and the nearby community of Robinson's, which is a lot of snakes. So I have tissue samples from 100 and some Newfoundland snakes from 3 different populations. And what I need to do now is get tissue samples from the native range of snakes to actually compare the genetic makeup of invasive and native range snakes. The problem is, this is the range of common garter snakes in Canada, which is a daunting task to try to get tissue samples from across that range. But parsimoniously, This is the closest area to Newfoundland in the native range. So this is Nova Scotia. There is a daily ferry that runs from Cape Breton, which is right here, up to Newfoundland. And also, if we look morphologically at the snakes, a lot of you saw my poster yesterday where I talked about this at length, but morphologically, the snakes that are found in Newfoundland are very similar color-wise to snakes that are found in Nova Scotia and New Brunswick, and which suggests that they're probably maritime garter snakes, so the subspecies Thamnophis, sirtalis polygulus. But this is a snake from Ontario, so there is that sort of potential for variation. It looks quite different from the snakes that are found in Newfoundland, but it looks quite different as well from what we think of snakes in Ontario looking like, right? This is a eastern garter snake. This is actually the Wikipedia image for eastern garter snakes. So that's, if you're not from Ontario, this is probably what you think an eastern garter snake looks like, maybe. Right now, I am a graduate student looking for tissue samples from Thamnophis, sirtalis, and TS polygulus. So if you have liver, blood, muscle, any kind of tissue sample from one of these subspecies in your freezer somewhere, and you're not one of the people I've already asked about this, hit me up. This is right now. Let me know. We'll talk about if I can borrow a tissue. So once I do that, this is my goal right now, but where am I going from there? The first thing is determining these behavioral differences using the actual source population. So if I'm just looking at random common garter snakes, it's not going to give me a lot of information about how the source population has adapted or was already able to like exhibit these tendencies like exploration and boldness. So to test defensiveness is one of the traits that I'm testing. I'm taking a video recording while I'm handling them to get sort of defensive behaviors from a reaction to a predator, basically. For exploration, I put them in this arena. I like to call it the rink. Actually, sometimes I call it the octagon. If I want to be really ominous, but it has this grid, so I put them in there and then I film what they do for 15 minutes, see how much they explore, and then I scare them a little bit and scare them into one of those hides that you can see. That's what these two things on the side are. These are optional hides for the snakes to use, so I scare them into one of those, and then I wait and see how long it takes them to come out after being scared by my scary hand. So that's my next objective for once I figure out what the source population is. And then my final objective is predicting the potential ecological impacts of garter snakes on Newfoundland's native ecosystem. So one thing that we've observed, this is actually one of the Newfoundland garter snakes here. This is a still from a video that a woman in St. David sent me of a garter snake actually eating a toad. So there's American toads all over the place in Newfoundland, and they're not native either. So that is very likely that that's a good prey source for these animals. So we're going to figure out if they're actually eating invasive species or native species. And then using the genetic data and the source population data, we can work towards finding a possible invasion vector and preventing more invasions. So the theoretical implications are looking at how this invasion fits with what we already know. And then learning what it tells us about how animals adapt to novel conditions. So this is IUCN data about various classifications of risk level to different taxa. And then zooming in on our friends, the reptiles and amphibians, we can actually use the data and the information that I'm learning from this PhD project and apply it more broadly to not just invasive species, but species that are experiencing changes in their habitat. So an invasive species is basically a species that's taken from somewhere and put in a new habitat, but anthropogenic modification is basically taking new habitat and putting it where the species are. So the adaptive features should theoretically be very similar. So we can actually apply this knowledge to conservation of other species that are threatened by human activity. With that, I think I'm running out of time, but thank you to a large number of people, including my supervisors, my field teams, all of these people in Newfoundland that helped me so much, and the people that gave me money, and mostly to the snakes for volunteering for this study. here's again my email address if you have tissue samples of these two subspecies. I'm on Instagram, and then this is a QR code if you want to check out more about snakes in Newfoundland. This is a link to the Facebook group that I have there. Yeah, how am I doing? Good? Perfect.
Thank you, Andrea. We have time for a few questions.
Awesome.
I guess my question is, I don't know a lot about what is in Newfoundland. I mean, I've been there, but given what I understand about post-glacial expansion, this kind of seems like maybe a thing that might have happened anyway.
Yeah.
So is it really, I guess, what are the consequences in Newfoundland? What are the major concerns? Or is this now, okay, maybe this is not a bad thing?
So the major concern right now is that they might actually be eating native species. It's fairly unlikely because garter snakes really tend to eat, like in the environment that they're in, there's so many of these inurans. There's actually multiple species of invasive inurans. So I think it's very, very, very likely that that's the major thing that garter snakes are eating. And in all of the communities where we find garter snakes, we find these huge exploding populations of invasive inurans. But there is some concern because garter snakes can basically eat anything that they can fit in their mouth, that they might be eating like at-risk birds or endemic small mammals, which there are not very many in Newfoundland. So the province is concerned about that as a potential ecological impact, but that's sort of what I'm going to find out with I'm going to be doing diet analyses to see what they actually are eating, and then we can kind of actually make some calls from there. But yeah.
I'm just wondering, how is the province responding to it? Are they collecting them, or can they kill them, or what's the deal?
Yeah, so the province is actually discouraging the public from killing snakes just because the public tends to do it not very humanely. But it's actually in my permits that I have to euthanize them. So that's sort of, and when the like anybody who works for the Provincial Wildlife Division encounters one, they're supposed to euthanize them using humane methods.
Thanks, Andrea. That's super interesting. Just a little story that I have. Oh, still on? Yeah, still on. I had a herpetologist who's an author of one of the field guides here come visit me on Vancouver Island in order to see a species that she doesn't have in her local area. On her way home, she's driving down I-5 to Seattle, and a snake starts coming out of her vents.
Yeah.
So it came from Vancouver Island, and now she was near Seattle. She went and parked and tried to get out of the car and couldn't find it. So I'm just wondering if through your Facebook settings, because you have a ferry, if you want to somehow find out how many people have maybe found one in their car or have seen one in their car.
That's a really interesting, and I've had nobody come forward with that information. I have heard stories, so sort of secondhand through a person that I know in Newfoundland. He knows a guy who works on the ferry, and he said he's seen them in the cargo hold in Port aux Basques. Yeah.
We can take one more question. We're trying to synchronize with the other session there. So one more question.
Sorry, I just also, you had that one dot on that one island. Did you go to that island?
Yes, I did.
That's kind of interesting.
We'll go back. Hang on.
I guess there's a ferry to there then.
No, it's actually, you can drive onto that one. If I go all the way back and show you. So, oh, okay, that's close enough. So up here, this is actually, this map is very simplified. So there is actually a causeway that connects this. This is a community called Virgin Arm, and you can actually drive up there. And that was a really weird case because somebody had hit that with a car. They posted a picture on Facebook. Selwyn and I beat it up there, trying to I texted the guy and I was like, "Can you please pick this snake up and save it for me?" And he was like, "Sorry, I saw it a day or two ago and like the crows got it." But I'm like, "Okay, well, we'll drive up there and see if there's anything else around." And that particular snake was very Eastern garter snake looking to me, which is really unfortunate that I don't have it. I do have the picture, but it was like very black with like very solid kind of yellow striping on the sides and on the dorsum. And then I found I talked to one person who lived across the street from somebody who had a pet garter snake at one point, and she was like, that might have been somebody's pet that escaped. So that maybe was a one-off. But I'm very keeping my eye on that area because, Yeah, it's a weird one. And it's like you have to cross 2 causeways to get there. So there's no way that a garter snake would go there naturally, I don't think. But yeah, that's a really good question.
Well, thank you very much, Andrea. That was great.
Yeah.
Our next presenter is Maya, and she's going to talk about belly in the beast.
Good afternoon, everyone. Thank you so much for that introduction. Today, I'm going to be discussing invasional dietary mechanisms and how they may have supported a newfound population. So invasive species are a globally pervasive threat. They are a leading cause of biodiversity decline worldwide due to their negative impacts on native populations, ecological communities, and the economic strain they cause. This is mainly due to loss of productivity, human health effects, and the high cost of preventing, managing, and/or eradicating invasive populations. A classic example we often think of is the cane toad. Native to South America, this toad has colonized over 1.2 million kilometers squared of habitat in Australia and has led to many localized extinctions, notably through the removal of apex predators, destabilizing food webs and trophic cascades. Similarly, brown tree snakes were introduced to Guam in the 1940s and have decimated the island's avifauna, leading to the extirpation of 12 bird species on the island and declines of over 90% for another 8 species. Unfortunately, within the modern era, and despite our increasing efforts to curtail their spread, we're seeing more and more invasive populations across the globe each year. This is due to increased human activity, ever-growing transportation networks, and anthropogenic habitat change. But as my handsome colleague was previously discussing, invasive populations don't actually have a very easy time. They form after non-native individuals progress through a series of invasion stages, whereby they must survive transport to a new location, survive within the new location, reproduce and maintain a self-sustaining population, and then expand their range beyond the initial site of establishment. At each and every point, there are abiotic and biotic barriers, or in this case, hurdles that the species must overcome. Because of this, it's no surprise that most invasions actually fail, especially after accidental introduction, because we just don't see it. Therefore, when species do become invasive, it allows us a very unique study opportunity. So I'm going to take us back over to Newfoundland. Within the herp context, Newfoundland is a very interesting study location. The eastern Canadian island has no native amphibians or reptiles, but now hosts the largest assemblage of domestic invasives in Canada. So I'll just walk you through it. I know we got a little bit before, but here it is. So in the 1800s, the green frog was first described, and then the American toad, wood frog, northern leopard frog, and western chorus frog were described in the 1960s. Both the northern leopard frog and western chorus frog were unable to overcome those abiotic and biotic barriers and are presently no longer on the island. In the 2000s, the mink frog and the maritime garter snake were first described, although we now know that the maritime garter snake was there for a lot longer. So it begs the question, how long have these guys been over there? And most recently, in 2022, eastern redback salamanders were described, maintaining a self-sustaining population in Conception Bay South. These are generalist predators that can form exceptionally high biomass and play an important role in nutrient cycling. Therefore, their most recent description has raised some eyebrows on how, why, and what are they doing. So, to understand this mystery, we must first understand what are some of the very first barriers a species must overcome. Getting enough and the right food is incredibly important, as they need to meet their energetic needs to survive and reproduce. This is where our work comes in. We wanted to understand how changes in their diet may have allowed this species to settle into island life. So, for this work, we looked at 2 main hypotheses, the first of which is the niche breadth invasion success hypothesis. This posits that invaders show a dietary shift to be able to capitalize on a wider diversity of prey in novel environments. By increasing the diversity within their own diet of an invading population, they can decrease the likelihood of competitive exclusion. Basically, by spreading themselves more thinly, they can take little bits of prey from native populations and not be completely outcompeted. So our first question is, how have they altered their diet in the new range? And we predict that invasives will be more generalist with a wider diversity of prey and increased evenness when compared to native conspecifics. The second hypothesis is the invasional meltdown hypothesis. This posits that invasions create positive feedback loops that create open niches, allowing for more invasions. It's a bit complicated, but it works alongside the first hypothesis, as it again minimizes the likelihood of competitive exclusion. Basically, the invaders come, and if they're an invasive invertebrate per se, they create an open niche for an invasive predator. When this new invasive predator comes in, they aren't competing with any native species, and thus they have a higher chance of success. So our second question is, are invasive prey a large component of their diet? And we predict that the invasive salamanders are eating a high proportion of invasive prey, both in abundance and in volume. So the data collection. 133 individuals were collected in the field in Conception Bay South. They were euthanized and then dissected, removing the stomach from the top sphincter to the bottom. So you can see here, let's just wait. We have our salamander, and the stomach has been dissected with a ventral line along their surface. The stomach is removed, cut open underneath a dissecting microscope, and flushed with 99% ethanol. The prey items are then unfurled, because as you can see, they're pretty clumped up in there. It's pretty crazy. And they're unfurled, and we organize them and identify them to lowest possible taxonomic level, grouping identifiable bits together. So if you have like 3 Hymenoptera legs, you're going to add it to like another individual, another leg, just so we're not inflating our results. We then posted any unknowns or ones we wanted more consultation on iNaturalist for community-based crowdsourced consultation. And all prey items were either identified as invasive or native using the 2020 Wild Species Index. For the native range, we conducted a systematic literature review where we screened numerous articles, but ended up screening 70 using covidence. We then went through all of published herp review with a controlled find function. Believe me, it took quite a few nights from 1967 to 2023. And we ended up compiling data from 14 studies across North America with over 19,000 prey items within the native range. So here we can see the native range of Plethodon cineris in light green, and we have our 24 native sites along there in dark blue, and then our 1 invasive site in orange. So what did we find? We found a strong dietary shift in the invasive salamander population from the native population. Mainly, proportion-wise, we found a decrease in the proportion of mites being eaten and an increase in the proportion of springtails, isopods, millipedes and centipedes, and beetles. We also found that our invasive salamander's diet is more diverse and even than native salamanders. So we did Shannon Wieners comparing both the diets and Pilus J. So you can see them on the top and the bottom there. So what about if we break down our invasive salamander's diet and just look at that? From that, we see that 36% of the invasive salamander's diet comes from invasive prey items, which is actually quite a large amount by count. But what about by volume? Volume-wise, we found that significantly more volume in the salamander's diet is made up of invasive prey items. This ended up switching, so by count, it was 1/3 was invasive, and actually by volume, it was 2/3. So this is on a log scale here. So what did we find? We found that because of the strong differences in diet, that the niche breadth invasion success hypothesis is supported. And this aligns with previous work done on African clawed frogs in their invasion to France. So they changed their diet when encountering the new environment. We also found a very high number and volume of invasive in diets, which suggests that the previous invasions of invertebrates to the island created open niches that our salamanders were able to capitalize upon. This has been seen before, as a global review of the American bullfrog found that their invasion has been significantly bolstered across the globe by the previous invasions of crayfish, which is pretty crazy that those two go hand in hand. Our findings, unfortunately, do not take into account the native prey, like the prey assemblage on the island, because the initial collection events were quite unexpected. And basically, they're like, there are these salamanders, we've got to go and get them. So we don't know if this is just a generalist predator eating what's on the landscape, or if they're selecting for these invasive prey items. So future studies should look at what is available, and ideally at the same time that these salamanders were collected, and run electivity studies. So are these salamanders actively selecting for larger invasive prey, or are they just eating what's around them? With increased urbanization and globalization driving an uptake in invasive species, especially in human-modified environments, it's incredibly important for us to understand the mechanisms facilitating these invasions, especially dietary mechanisms. By breaking down these mechanisms, we can better predict, manage, and prepare for increased invasives in our communities. I would like to extend my deepest gratitude to James and Julia for supporting this project and the rest of the Riley Lab. Please follow us on Instagram, RileyLab_MTA, as well as the funding bodies that supported this work. I'd also like to thank the landowners whose work this was done upon, the Newfoundland and Labrador's Wildlife Division, and especially the iNaturalist community, because they really supported this work. I'd also like to thank all those scientists who came before me, whose work I was able to build upon, and especially those who published and shared their data with me, because open science is the future of science. And I'd like to leave you with this, and I'd love to take any questions.
Excellent. Thank you so much for that presentation. The question I have is, if we find out in the future that they are targeting or mostly just eating the invasive species, right?
Yeah.
As we all know, we're seeing herpetofauna decline globally, right? If they're not causing any damage to the habitat there, do you think it's possible they could just be taken off that invasive species list, especially because they're from the province right over?
Absolutely. So I think it's pretty interesting because it's only in 2022 that these were removed. The province, same as with the garter snakes, it's like in the permits that if you come across one, you need to euthanize it. But the province is looking at other options, you know, like it is important. Like we said, like it's crazy. Most of the talks here are about conservation, and here we are taking them out of the environment, which, yeah, it's interesting to see what will happen next.
So I'm wondering, this is more of a general question for you and Andrea and everything. Is the government of Florida North, which is now Newfoundland, are they looking broader? Are they thinking like, we don't want, you know, I was joking, but we don't want to be Florida. And so is there a ramp up of protective measures, or is it like throw up your hands or science goes slow like it always does?
I think it will go slow. As of I know right now, our lab is the only ones really looking into this. So it'll be interesting to see maybe if MUN with Sean Boyle start looking into stuff, or, you know, what they do in the future. You should, follow us on Instagram, because the crew actually this summer found some more species as well. So there's way more than we were expected. It's interesting. Newfoundland doesn't really seem, at least from my experience, to be like really talking about this fact. Even like they have some museums and some toads in the museums, and they don't really say anything about the fact that they're not native. So I do wonder if maybe it was just a previous unawareness or just like lack of science research.
You don't get a sense that the current government is now ramping up and saying we've got to develop a protective space around the island.
We'll have to see. I mean, it's different. I know they don't like scan you for any frogs in your pockets when you like go over the border, but maybe they should. Yeah.
Just a brief question about the invasive invertebrates. Is anyone looking at those, and can you use the salamanders to sample them and maybe find some new ones you didn't even know about?
Well, I have another slide. So I'm the first person to record this invertebrate in Canada, which was a pretty big deal because they're from Europe and highly invasive. So it's a harvestman. And it's like called a pearlback harvestman or something like that. And I found that, as well as another weevil species that has only been found once before in Canada. So it's pretty exciting to like post these things, and people are like, oh my God, read this paper. Yeah, but it's definitely a possibility and easy to do. Thank you.
So I'm just curious, all these frogs that were introduced in the 60s and others,
Yeah.
do they have any idea how they got there? Were they accidental or anything intentional?
We're not completely sure. There are varying accounts, so maybe we should talk about that without a microphone.
Our next speaker is Nick Cairns, and he's going to tell us about undocumented species in Alberta.
All right. So speaking of invasive, the Riley Lab has no right to be this good this fast. It's totally unacceptable that you guys are this talented. So that was fantastic. All the talks have been great. So what? What? Oh, yeah. Yeah, look at that guy. Yeah, no, once a year I dress up. This is the one time. I have my other clothes. I will wear this for about another 20 more minutes before I stain it. So, yeah, thank you for, as ever, inviting us to wherever you guys live and having all of us invade your towns. One day I hope for people to invade my town, although the herping in Edmonton isn't quite the same as up here. But yeah, I just wanted to first off say thank you to Chris Kendall, Sandy Robertson, Sherry Monk, Andy Didiak, a couple fantastic people in the prairies who have given me a lot of information on some of the species as I'm getting to know a slightly more northern habitat than I'm used to. But I'm going to try and talk about richness and distributions in Alberta. And hopefully I can convince some of you to give me your locations near the border of Alberta so I have places to go and search in the future, because I don't like being inside. So how could we miss something in Alberta? It's a rich province in a rich country, pretty high human density. It's got armies of consultants, armies of biologists roaming all over the place looking for species at risk so they can build things on top of them. So we've already got a book. How could we miss something? But the thing is, inside the book is a hint that maybe we already did miss something, because my favorite part of any guidebook that has the guts to put it in it is the page that says what may or may not be here. That's my favorite page in any guidebook. Most of them don't have them. This one does, because it's a good guidebook. So this is proof that we can miss things. This, everyone know what this is? Most of the British Columbians will know this, although you'll be slightly wrong by a subspecies. This is an eastern yellow-bellied racer. This is a hatchling. But up until 2014, these were not listed in Alberta's fauna. There was a certain group of people that knew they existed, and they just didn't bother to tell people, or someone didn't bother to write it down. Whatever, we didn't ask too many questions and no one got upset, but it's there now. Anyone that's seen an eastern yellow-bellied racer knows they're a fairly large diurnal snake. They aggregate for brumation. They're not particularly hard to find. Now, everyone that's visited grasslands and I said, oh, you'll find one, no worries, and you didn't, they're not that hard to find. I gave you everything you needed. But if we can go until 2014 and still miss eastern yellow-bellied racers, what else can we be missing? And there's a good portion of southern Alberta that actually has very, very low human density. So there are two main areas that I think are underexplored on the Alberta side of the line. The southwest, this portion of sort of from Crow's Nest Pass to Waterton Provincial Park, that is conifer forest, high peaks, low valleys, lots of fast moving cold water. It's a complicated area to survey. I know there's been lots of surveys, especially in Waterton, but that far side next to the BC border, that's a hard area to get to properly survey with equipment. And there's a lot of species that occur in Montana and BC that we don't have in Alberta, and I can almost guarantee you some of them are going to be there, especially if it means I get to do it for the next 25 years. So the first one that I think we'll be able to find, and that I would really love some British Columbian influence on, is Rocky Mountain tailed frogs. I know there's been a really good survey program going on in Waterton. They haven't found them yet, but their programs are are busy and they often don't get enough people to get the work done all the time, and they can't leave the national park boundaries. I think a good portion of you now, after a lot of really fantastic talks, some of which have mentioned tailed frogs, are going to be able to tell a tailed frog, but just in case you're wondering, vertical pupils, no tubercles, ascaphus. Their feet are relatively uninhibited by tubercles or spades or anything. Just beautiful, smooth, flat feet. The males also have a penis, so that will help because they're the only frogs that do. There's also not that many other frogs in that area that you're going to mistake them for. The tadpoles are way more likely to be seen than the adults. They have these big suctatorial mouth parts. They cling to smooth rocks and gravel in fast moving streams. They often have a little white spot on the tail, and often you'll see distinct cohorts by age group because they take so long to metamorph. The type of habitat you would find these in are these sort of enclosed, open, usually fishless stream, or enclosed streams, usually fishless. Often they'll be cover. You do find them in these bigger streams that are open as well, but they're usually quite cold and moving reasonably fast. And you can see in Montana, they go, there we are. They go right up to the border. Well, this is roughly the line. This Flathead Glacier County is roughly the line where Alberta starts. And so I'm really hoping one of these days we'll stumble across one of these things. So if you are from British Columbia, hint, hint, and have a bunch of cool observations from the east coast of the province, I would really love to talk. The next one that I do hope we find, and one of my favorite species from growing up in BC, is a rubber boa. This is also another fairly hard to mistake species, usually unpatterned, green to brown, dog turdish, and with a bumpy sort of unadorned tail, large head-like tail. Really, really cute snake, genuinely the cutest snake. I mean, sharp tails might give it a run for their money, but rubber boas are very cute. And they're not found right to the border on the Montana side, but up in BC, going up further north, they actually get much closer to the border. So I'm looking for sites where people would think that maybe a low mountain pass or an area that might be suggestible, and then I can go and look for talus slopes and try and pull one of these out of a rotten log at some point, hopefully. The next area is a bit closer to home for me. I've spent most of my adult life in southwestern Saskatchewan. I'm pretty used to the sort of Palliser's Triangle dry grasslands habitat. So down here is where I've called home for a long time, and it's a lovely place to go looking. And one of the best parts about it is there's no people. And so it's also relatively underexplored. This area where the Milk River crosses is an interesting mixture of heavily private land that access is difficult sometimes, and large provincial properties that access by vehicle is not allowed. So if you want to go look for something, you have to carry everything in and carry everything out on your back, which is fantastic because most people won't do that. So the species that I'm going to be looking for here are some of my favorite species from looking in Montana. The Woodhouse's toad comes pretty close. The Woodhouse's toad is a very typical Anaxurus buffonid toad. The tadpoles are very, very typical of buffonids when they're small, small black tadpoles. They do have often gray bellies with some gold flaking, which does allow you to distinguish them from the other congeners in the area. As they get older, they start to develop the adult coloration like this one from Montana did. And then the adults are very typical, nice white line down the back, almost symmetrical green markings, but the best way to tell them apart from their, best way to tell them apart from their congeners is the cranial crests. So when you're looking at a Woodhouse's toad, you'll see the cranial crest here is sort of L-shaped. It comes in contact with the parotoid glands, and it doesn't meet at the nose. If you look at a Great Plains toad, it's V-shaped, and the elements are lumpier, and the postorbital crest doesn't touch the parotoid glands. If you look at a Canadian toad, these little weirdos don't have postorbital crests really to speak of, and they have a giant boss right in the middle of their eyes. This is an exceptional individual. That boss is overdeveloped compared to what you'd typically see in Alberta. That's a Manitoba individual. But they don't have that postorbital crest portion. So their parotoid glands remain free and uninhibited. So when you were looking at these, if you do ever see a toad in southeastern Alberta, please take a picture of the top of its head, because the side of the toad is often unhelpful. Also, the thing to remember with Anaxyrus is they can all pretty much breed together. So you get these interesting hybrids. The exception being the Great Plains toad, which doesn't seem to hybridize. I'm going to go back a little bit just to see where you can find them. So this is where the Milk River crosses. So that's the Saskatchewan border's about there. So where the Milk crosses is an excellent spot, because this is a species that tends to stick to valleys that are dominated by willow and cottonwoods. Those are the types of habitats where this species is found. If you go too high up on the uplands, they tend to get replaced by Great Plains toads. But where they're down in these valleys, they're extremely common in places. And so following these valleys just makes sense from a biogeographic perspective. In Montana, they haven't been found right on the border yet, but largely that's due to the fact that this is mostly private land in this area. So surveys are relatively limited in terms of what the state of Montana is going to be able to do. The final species is my biggest long shot, but the one I want the most. I've been hunting milk snakes in Montana and Saskatchewan for 20 years now, nearly. I've yet to see one. Milk snakes in the northern Great Plains are quite difficult. They're really only active for a couple weeks in the spring, and the rest of the time they're in rodent burrows deep under glacial erratics or under big flat stones in river valleys. They are pretty hard to mistake for anything else, except for maybe escaped pets. Of course, most of the people in this part of Alberta don't keep snakes as pets. So it's usually unusual to see a bright red and white snake. These are both from fairly far north. Thank you. I'm going long because I'm enjoying myself. So these are still fairly far. I do have records from Saskatchewan that sound like they're reasonably well founded. They came from my wife. I tend to believe her. There are some really excellent records from very close to the Alberta border, so I'm looking forward to verifying those. They're unverified. They're not on the Montana Natural Heritage Program's website yet. But we'll move on to the next one, because the next steps are kind of fun. I've been seeking leads, which is what I'm doing here. So thank you for listening to this talk, especially if you've listened to it before. I'm going to be putting out posters in the local communities, because local sources are often the best sources, and doing presentations for school groups and things like that. Then once I've assessed some priority sites, we'll be doing ARU and foot surveys. We did a little bit of foot surveying this year already, putting out cover boards and doing some aquatic surveys. Some of the early observations, we work with some fantastic entomologists as well. We've added 2 species to the province, one genus of tabanid. If it hadn't been for some pesky herpetologists in Saskatchewan, it would have been the first time anyone's seen it in Canada. And then we also added about 50 kilometers to the range of eastern yellow-bellied racers in Alberta. So we're finding some things. We just haven't found any on my list of 4 there yet, but we'll get them. Still doing spadefoot work. The painted turtle work that associated with the Milk River is getting really scary, so hoping to ramp that up, and still doing my horned lizard work, so happy to talk about that stuff too. I think I'm low on question time, but if anyone has a quick one, I have to take it.
So Nick, do you think that there's any use in doing some modeling to just see where natural range expansion is going to happen with climate, to try to figure out which candidates are most likely to naturally move up?
Yeah, I mean, in the northern Great Plains, the modeling would be tricky because it's such a dynamic area to start with, especially in this sort of rain shadow part. It's certainly a valuable exercise. A lot of these are close enough to the border that I'm not not expecting that it's new migration. I'm just expecting we missed them. But yeah, I think it's always a valuable exercise just to understand where things are going to go. There's only so many like relatively logical biogeographic routes though, and that's sort of where I've been focusing. But yeah, like if there's students that want to do that, I'm all for it. I'll get as many data points as I can find. The Montana Natural Heritage site is a fantastic site for those sort of data points though too. So yeah, they have done some of that already.
So to conclude this session, last but not least, is Mark Thompson, and he is going to talk to us about abduction.
Yes. Hi, everybody. Just kind of anecdotal connection to the last talk. I was a member of the Anthony Russell Lab, and I was just realizing I had done a lot of surveys down on the Crow's Nest Pass area. So, Nick, maybe I'll talk to you afterwards. So I'm going to be talking about a book chapter that I've been working on, or that I've written. It's part of the amphibian biology series, and I was invited, and it was a great honor to be invited to work on a chapter within the status of decline of amphibians. And so we did chapter 5, and so this is my authorship, co-authorship group. Dr. Jamie Fenneman, who's in the room with us, helped to write on some of the geography. Dr. Sarah Ashpole, who is also maybe present in the audience here, she's been around, had helped with the Okanagan amphibians. My first graduate student, Candace Huckster, who had helped write on her work. She's done a poster here on tracking of western toads. And then I've invited 2 members of Nak'azdli Whut'en, who I've been working with for the past 20 or so years. Greg Crooks is a forester, and Tina Erickson, she's a Lisuliu Frog Clan member and an elder who they put together a box section in the book talking about Indigenous values related to amphibians, and wrote about the forgotten ones in the community. So in the book chapter we put together, I did a lot of mapping. This is kind of what I do for work. So I developed some maps where we looked at doing a lot of the bioclimatic zones, just to kind of give some nice figures. This was the area that I was invited to work on, was in the western Cordillera areas, including the taiga in the north. And then so I put together the physiographic provinces. And so the Nak'azdli Whut'en takes about 2.5 million hectares in the central part of the province from, well, that's not the thing. So from the Chaco lowlands up towards into the Casihiro-Uminica Mountains. So in the book chapter, we did I decided, well, I wanted like lots of maps and lots of figures. So we did some habitat modeling. I collected all the records that I could locate for western toads, wood frogs, spotted frogs, long-toed salamanders. We took some other distribution models that have been published and did a comparative analysis from other published studies, and looking at some of the records that I did, and compared some of the modeling techniques, where I had looked at some different environmental covariates to be able to put that together. And so, yes, so we just were looking and interested in trying to be able to do an evaluation of where the populations were, to be able to do an assessment. Population status was, well, I wanted to know where the populations might be. And so that was what we did, to be able to set it geographically, to be able to dial in the chapter and what we're talking about. And so we were asked to put in a lot of photos, and I've been doing a lot of work up in the northern parts of the province. And this was a great opportunity for me to be able to just kind of, broadcast some of the work that I've been doing, where we've been doing some die tracking of, keep on hitting the wrong button, sorry. And this one actually, just anecdotal, is really kind of neat. We found a western toad poop nest. There's a scat. So we tracked this toad, and so we can talk to people about that later. We see lots of signs of mortality. This one here is actually interesting. I'll talk a little bit in my talk about blowflies and cases of myiasis, and I did a little paper on that. And I actually got some people that have been sending me some new photos on blowflies that they found in BC. So this is something that's exciting. We're also finding water molds, and we talk about a lot of this in the chapter, and in particular about forestry road ponds. And so I'm going to flip a little bit here talking, I'm interested in the relationship between Indigenous knowledge and scientific, linking scientific and Indigenous communities together. I've organized a couple workshops on this down in the States. And so I brought together systematics, and this is a colleague or a friend of mine who is a curator of polychaetes, which kind of seems an odd connection to amphibians, but Dr. Fitzhugh has been a friend and somebody that I've been inspired by. And we'll talk a little bit. When he started studying, he was inspired, and his supervisor said, well, don't really go too deep into polychaetes. Study the philosophy and the epistemics of what science is and how it works. And that's what he's instilled in me. And so he's been publishing, and it takes a long time to try to really dive through his papers. And he's been largely inspired by Charles Saunders Peirce, who also inspired John Dewey, who many people know. It's just a very common name. And so this is what we're going to talk a little bit about, how that relates to conservation. And I talked about that in the book chapter. And so I wanted to bring up, when I'm thinking about trying to do a status assessment or thinking about conservation of amphibians. Well, I'm interested in causality, and we know that that's important in conservation. And, here's a highly cited paper with Charles Krebs that has talked about that. And this is pretty much one of the fundamental aspects of science, is understanding about cause-effect relations. And if we can understand the relationship between cause-effect relations, Stathos Philos is a great philosopher to read on this. He's written on Charles Peirce, who I'll introduce later as well. But if we know the causes, then we can actually develop some recipes that are predictive, and that's really what we're trying to do in conservation. We understand the mechanisms and the processes, and then we can plan out and ahead and do better. And we can thank this handsome gentleman, David Hume, for understanding causality. And really, it's as simple as he described it. Billiard balls are pretty much what we're dealing with in conservation, something about the size of a tire and a toad that they smack into each other. And he said that you had three kind of pieces to causality. You had spatial contiguity, that, of course, you have to come into contact. I don't think that quantum entanglement really matters too much for conservation. And temporal succession, cause has to precede effect, and spatial, and the constant conjunction betwixt cause and effect, as he put it. It's just kind of, well, we see this happening here, those cause effect tends to happen in this situation. I see that over here. So it's repeated around, and that's the constant conjunction that tends to happen. And he said, well, the problem is that we can put these three things together, but there's no extra ingredient that I can say that is a cause. And so that spurred on much debate over history and the philosophy, and I'm not going to go into depth about that, other than it's relatively easy to put it together. That causes are really things that you can see in organisms that are smacking about. And I just wanted to highlight here that we know that correlation does not mean causation, but it implies it, and in most cases it's just an untested cause, and I want to bring up that point later. And so I wanted to introduce to the world of conservation the idea of abduction. Well, many of you may have heard about induction and deduction. Well, that's the process of testing. And then so we want to know about abduction, and this is Charles Saunders Peirce, who really made it his life's work, and maybe it's the deeper philosophy of hypothesis. And so when I'm out in the world and I observe the effects that are around me, well, it's an effect. What I do in abduction is I infer a cause for that effect. Observing and perceiving things, I remember it. It causes my memory. I communicate that to other people. So I'm making observation statements, and it's an extension from the effect that I witnessed in the world. And that's that is the key entry point to all new knowledge, and that's the entry point to science. And so this is where I'm going to link this into the way that we relate back to Indigenous knowledge. And our brains are good abduction machines because we can understand causes, as you can see and hear, that we can correctly perceive and guess that, well, that cat did not break that table or chair. And I'll just leave this here for a moment because I think Charles Peirce described abduction best. And I wanted to bring this, and this is part of the philosophy that Charles Peirce, or not, Kirk Fitzhugh has introduced to me. And a lot of the way we talk about species is we talk about them as though they are causal agents. They split, spread, have distributions, have perimeters. But it's really the case is that what we perceive are organisms, and species are the inference, that they're an abstraction, that they're part of a proposition and they're explanatory hypothesis. And as such, we rarely test them. And this is not a new idea. In fact, Charles Darwin would have accepted that kind of idea about species. And you can, I got this from Ernst Mayr, who I'll talk about later. Charles Bessey, a botanist who has a bust. So obviously he was an important botanist. But, you know, I think that I just grabbed some three key points, and he gave us some warning here, you know, that species have no actual existence in nature. What we have are organisms, and species are an extension of our inferential products that we make about organisms to provide an explanation for the effects that we observe in nature. So let's look at it from the great biologist Ernst Mayr, who helped to really dive into how causality works in ecology and systematics. And so, you know, when we look at an example of the biological species concept, it's really what not the species concept, but the biological species hypothesis, that it's vague on causality. We have populations that are split historically. They're reproductively isolated. So we have causes that happened historically, and they tumble down through the great chain of time, through reproduction, heritability, and then we observe the traits and the characters and the organisms at present. And so that's the inference that we're making, that we're providing an explanation for that history. So it's actually not a thing. It's not an entity in the world. So Ernst Mayr gave us this idea of proximate historical causes. Proximate causes are those that occur within the history, the lifetime of an organism. An example here is long-toed salamanders. I often find them with autotomized tails. You flip over a rock, you'll find a tail, and it's squiggling away as they quietly sleek away. I've done a little paper here where a photographer captured about the kingfisher eating long-toed salamanders. And here's an example of an abductive inference. Well, I would think that that's a toad, or a bird's beak that bit into the tail. So that's a proximate cause, and historical causes are those that occurred prior to the lifetime of an organism, as we would have in a species explanation hypothesis. And so I mentioned the blowflies. And so when I was working, of course, you see a hole in the side of the toad. This was my first observation. Well, that's not what you would expect. But so that's surprising. And so we got these maggots. And so piecing together this story of understanding the relationship between these caliphorid flies and parasitism on amphibians, that it's an interesting story in terms of abduction. So I put, that I caught a fly with its pants down. So a couple of weeks ago or a month in August, I saw this fly walking on the backs of toads, and I keyed in. I was like, wait a second, that's the fly. And it's a nice little fly, actually. It walks around on the toad, and I picked up the toad and would fly away onto my hand and walk on my hand, and then would go back onto the toad. And so it was so easy. I just picked it up and put it in a bottle in a jar. And so we're working on that now, and I'm getting some reports from other people as I put this out, notification on these. But I just wanted to, I've got some points here that are highlighting what abduction is. And if you don't know what abduction is, I just wanted to invite people that are in the conservation world that we really need to start to understand that, because it really ties into natural history and our knowledge in general of how we are understanding and interpreting the world around us, and it has connections to our knowledge in general and how we're doing conservation. And so, so now, I've talked about species. Species are important as a hypothesis, providing, allowing us to key into the patterns of nature. But populations are really where causality is happening. It's within the local spatial contiguity. And so, in the book chapter, I've asked for many years, what is a population, and how does that work for us in conservation? And one of the challenges is that, when we're trying to study populations, it's very challenging to find long-toed salamanders and to do a study on them. you're looking at 20 years, before you're going to get a really well-funded study to be able to find this kind of trend and change and some kind of effect in the population prior to a collapse. And, one of the problems is, that we're doing inferences in long-toed salamanders from samples that are actually not within the geographic area. And we've extended an inference of security without actually sampling. And so one of my experiences that I see up in northern BC often is these dried wetland areas with, these are western toad smears of dead tadpoles. And it's a very common occurrence in forestry habitats. So I asked the question, what are the fixes and causes of amphibian declines? And I just wanted to point out that what is relevant is our primary observations of the organisms. And we need to understand the fundamental link between our observations of organisms and the inferential products. So the argument from Kirk Fitzhugh is that species are just hypotheses and that they are explanatory accounts for some history. And I'm just going to close off here just with looking at the relationship with Indigenous peoples and the entangled bank of nature that you see that they've illustrated in their historical artwork, and how we could link together and thread together the knowledge of science with the work of Indigenous peoples. And I think that abduction is the thread that can link our communities of knowledge together. Yes, thank you.
Thank you very much, Mark. That was really interesting. Unfortunately, we don't have time for any questions. So, but are you going to be around here so if people have questions?
Yes.